Good morning, everyone. This is Group 3 presenting on Euglenids. If you ever find yourself at any wetland area, a marsh, swamp, bog, or mire, it's certain that you're going to encounter a group of various types of euglenids, a type of distinctive, primarily unicellular algae. Some general characteristics of euglenids are as follows. They are single-celled organisms that are mostly autotrophic, but they can be heterotrophic. They are both free-living and parasitic. They have one or two flagella and have no cell walls, but their plasma membranes are strong and sturdy. They share characteristics of both plants and animals, which had led them to be classified as both at some point, but are now exclusively algae. Euglenids have their main habitats in places with an abundance of decaying organic matter. So apart from the locations mentioned before, you can also expect them in marine or brackish sand and mud flats characterized by decaying seaweeds or organic contamination, farm ponds, dipteran larvae hindguts, and even the rectums of tadpoles. Cell biology. The euglenoids, also known as euglenids, euglenophytes, or euglenoid flagellates, are the earliest known divergent or most ancient group of eukaryotic algae. Only about one-third of the known genera possess green pigmented chloroplasts, and of the rest may contain colorless plastids, and others lack plastids altogether. However, even the latter have typical eukaryotic mitochondria, cytoskeletal systems, 9+2 flagella, and endomembrane systems, including Golgi bodies. A number of euglenoid genera are phagotrophic, which mean they feed upon organic particles and consequently possess cellular organelles that are specialized for capture and ingestion of prey, including bacteria and small algal cells. Some euglenoid predators are indiscriminate feeders, whereas others specialize in diet, like feeding only upon selected diatoms. In response to changing environmental conditions, euglenoids may form resting cysts. Cyst formation involves loss of flagella, increase in the number of pyromyelin granules, swelling and the rounding of the cell, increase in the number of mucilage bodies, and deposition of a layered mucilaginous wall, consisting primarily of polysaccharides. It has been suggested that this is a survival response to low nutrient levels in the environment. However, this does not mean they undergo a period of hibernation, as it has been observed that euglenoids may sometimes continue to undergo metaboly, rotating within the confines of the cyst wall. Euglenid motility. The euglenid's mode of motility relies on by means of one or more flagella. These flagellar features are one of the earliest characteristics that earned the euglenid's taxonomic criteria. In some forms, such as the common and familiar euglena, only one flagellum emerges from the flagellar pocket. The other remains very short and is typically not visible. Other euglenoids have 2 emergent flagella, and some records of other few euglenoids have 3 to 7 emergent flagella. In addition to swimming, euglenoids can move by cellular undulations, known as metaboly, peristaltic movement, or euglenoid movement shown in the slide. These movements are readily recognizable and unique to euglenoids. Helped by the flagella, the cytoplasm appears to accumulate at one end of the cell and then is rapidly redistributed, resulting in forward or backward cell movement. In addition, cells can flex from side to side. Metaboly is enabled by the highly flexible and plastic pellicle. Pellicles are a type of cell covering that are up to 70 to 80% protein, and then lipids. Within its taxon, the pellicle is one of the euglenoid's most diverse morphological features. They exist on the organism as proteinacean strips underneath the cell membrane, supported by dorsal and ventral microtubules that can slide past one another, which result in metaboly. Euglenid reproduction. Although euglenids share several common characteristics with animals, which is why they were originally classified as so, sexual reproduction has never been observed in euglenids. Euglenids utilize asexual reproduction in the form of binary fission to propagate and by way of longitudinal cell division or longitudinal binary fission. Euglenoid diversity.
Phacus longicauda. It belongs to the phylum Euglenozoa, class Euglenoidea, order Euglenida, family Euglenaceae, genus Phacus, and species Phacus longicauda. It appears to be mostly green, leaf oblate in shape. It has rigid large cells which is approximately 70 to 160 micrometers long with long caudal prolongation and a flagellum of about 1/3 to 1/2 its total body length, which is approximately 55 to 90 micrometers. It also has a prominent stigma with many chromatophores, discoidal, paramylon body central, and a pellicle that is longitudinally striated. Lastly, it appears to be flat on the substrate while slightly twisted when swimming. Trachylomonas hispida. It belongs to the phylum Euglenozoa, class Euglenoidea, order Euglenida, family Euglenaceae, genus Trachylomonas, species Trachylomonas hispida. It appears to be yellowish to reddish-brown in color. It has an ellipsoidal lorica which is approximately 15.3 to 24 micrometers wide and approximately 26 micrometers long. It is uniformly and densely covered with short, sharp conical spines with an apical pore that is colorless or a short, slightly elevated pore. It has a flagellum that is approximately twice the length of the lorica and a cell with 8 to 10 plate-like chloroplasts in which usually with a pyrenoid but sometimes without. At a young age, it forms delicate and net-like envelopes, while at mature age, it already has a thick loricae with small pores.
Another species of Euglenids is Discoplastis spathyrhyncha. This species is from the phylum Euglenozoa, class Euglenoidea, order Euglenida, family Phycology, genus Discoplastis, species Discoplastis spathyrhyncha trimer. So these species are large cells that are usually greater than 65 micrometer with long tail on average 10 to 12 micrometer. It expands in the center during the euglenoid movement. Its shape is similar to a ballerina's tutu skirt. So as seen in the image labeled S, during the euglenoid movement, the center expands and shaped like a ballerina's tutu skirt. Its cell is cylindrical fusiform and it's very metabolic. Its anterior end elongated in a snout-like manner and diagonally cut. Its posterior end is a wedge-like narrowing and terminated with a sharp hyaline tail. Also, it has monomorphic paramylon grains that are oval or short rod-like. These characteristics are seen in the image labeled Q and R. Euglenoformis proxima. This species is from the phylum Euglenozoa, class Euglenoidea, order Euglenida, family Euglenaceae, genus Euglenoformis, species Euglenoformis proxima, M.S. Bennett and Trimer. So as seen in the image labeled D, it is fusiform with 52 to 90 by 13 to 25 micrometer and it is slightly tapered. It is spindle-shaped, rounded at the anterior end with a conical posterior part. It also has monomorphic paramylon grains that are small, rod-like and oval. Its periplasts are spirally striated. Also, its cell is metabolic with flexible pellicle. These species are free-living cells, solitary with one emergent flagellum when swimming.
The species Eutraptiella brarudi comes from the phylum Euglenozoa, class Euglenophyceae, subclass Euglenophycidae, order Eutraptida, family Eutraptidae, and genus Eutraptiella. Its average size length is between 64 to 115 micrometers. It is a solitary life form. Its geometric shape is rotational ellipsoid. Its cell is with 2 or 4 flagella. Its pellicula with broad stripes, and its chloroplast radiates from 2 paramylon centers. The Momorphina pseudonordstedti comes from the phylum Euglenozoa, division Euglenophyta, class Euglenophyceae, order Euglenales, family Euglenaceae. Its cells are napiform, nearly spherical but with a long, straight, sharply pointed caudus. It's broadly rounded anteriorly periplast, forming an envelope widely separated from an elliptical protoplast. The periplast spirally striated, paramylon bodies not observed, Chloroplast numerous, ovoid, indistinct disc. The size of its cell is approximately 19 micrometers in diameter and 36 micrometers in length.
Colacium vesiculosum. It belongs to phylum Euglenozoa, class Euglenoideae, order Euglenida, family Euglinaceae, genus Colacium, and species Colacium vesiculosum. Colacium vesiculosum is characterized by having a cell body that is attached to another surface by a gelatinous stalk emerging from the anterior end. Its cell body is also ovoidal or spindle in shape with a round posterior end, and is generally 19 to 30 micrometers long and 10 to 12 micrometers wide. It has a locomotive flagellum that is about 2 times longer than the cell body. Its chloroplasts are generally 5 to 10 in number and are saucer-shaped consisting of a pyrenoid, while its nucleus is spherical in shape and measures 6 to 7 micrometers in diameter. Strombomonas fluviatilis. It belongs to phylum Euglenozoa, class Euglenophysce, order Euglenida, family Euglenidae, genus Strombomonas, and species Strombomonas fluviatilis. As in all known species of Strombomonas, S. fluviatilis is a freshwater algae. It is generally 28 to 38 micrometers long and 12 to 17 micrometers wide. It is also reported to have a cylindrical color, smooth surface, and a slightly brown lorica, which is soft rather than brittle. Its lorica is largely unmineralized and thus can change shape during metaboly. Aside from its slightly brown color, the lorica of Strombomonas fluviatilis is also characterized to be broad, spindle-shaped, or ellipsoidal with a slanted opening, as shown in the
Euglena
figure.
viridis. It belongs to the phylum Euglenozoa, division Euglenophyta, class Euglenophyceae, order Euglenales, family Euglinaceae, and genus Euglena. Moving on to the morphological characteristics, most Euglena exhibit a fusiform shape. However, for this organism, it is specified to be 40 to 80 micrometers long and 12 to 20 micrometers wide, which allows them to swim rapidly. It also helps that their anterior end is rounded so that there is less drag. Their chloroplasts are more or less band-form, with the nucleus closer to the posterior end. And lastly, it has a very distinguishable stigma found at the anterior end, appearing as an orange dot in this illustration. Cryptoglena pigra. It belongs to the phylum Euglenozoa, class Euglenophyceae, order Euglenales, family Euglinaceae, and genus Cryptoglena. They appear to be almost symmetrical and oval in shape, with lengths ranging from approximately 14 to 17 micrometers and width ranging between 6.77 to 13.55 micrometers. As you can see from the illustration, an emergent flagellum of almost body length is identifiable, with a ruby red stigma at the right side from the ventral view. So we could assume that this photo is the dorsal view of the organism. It also has a pellicle like other euglenoids, which helps keep the integrity of its form. Lastly, it has a distinct 5 to 6 membered ventral root extended anteriorly.
Euglenids in ecosystems are well known especially because of their association with increased levels of dissolved organics, which puts them to use as environmental indicators of such conditions. Environments high in decaying organic materials suit the need of euglenids because they all require vitamins B1 and B12, which are released by the activity of associated microbial floras in which euglenids have been used to bioassay. Euglena gracilis, the most studied species, is utilized in research involving the improvement of food nutritional value for human consumption and livestock. Further researchers say that it has potential to serve as biofuel and decrease greenhouse emissions in the near future. A particular species of Euglena even produces a toxic alkaloid called euglenophycin, which is capable of killing fish and other algae exposed to large concentrations of the toxin, and at the same time have anti-cancer activity in vitro. To think that a species with all this potential is usually known as pond skunk. Thank you for enjoying our presentation.